can run 5 minutes. Okay, thanks everybody. We're in this room because it is Dean's Honors Day, and they're using our usual room. I have the usual couple of important announcemements. The first is I'm reminding all the faculty in the room that next week at group meeting is a third-year talk day, and the one after that is a third-year talk day. So everyone should come, but faculty should come or they'll be shocked. Heather wants me to remind everybody that they need to do flu shots. If you have not done a flu shot and you haven't done your flu shot by November 1st, you will regret it. So get your flu shot done so that the office team does not have to nag you relentlessly. Okay, so I have 3 wins I want to mention. I'm going to start with, where's Paul? Where's Shane's lab? It's embarrassing when they're not here, right? Well, okay, so Paul Frizzell and Shane's lab published a paper this week in Nature Neuroscience. So we can that's amazing. And I've got 2 large conglomerate papers I want to mention. I mean, it's like everybody in the departments on these papers. Kristen, Hannah, Dina, Paul, Mercer, Yunlu, Kayla, Kolchi, Shane, Shopik, and Jeremy published a paper in Cell Reports. That's pretty cool, so many people. And that wasn't enough. They have a second paper in Cell Reports, but this one is Yunlu, Francesca, Hannah, Samantha, Kayla, Christina, Hassan, Niels, Kathy, and David, also in Cell Reports. It's amazing. It's so nice to see so many people from 3 different labs collaborating, 4 different labs collaborating. And then I'm going to call out one accomplishment by a faculty member, which I know we never do here. Jaida and Mariah Thomason both got honorable mentions as outstanding mentors of the year. That's thanks to you guys calling out their hard work. And so I just wanted to congratulate them and thank you all for calling out their hard work. Yay. Okay. Where did Winnie go? Oh, there you are. Okay, Winnie, it's all yours.
Thank you.
Oh, and it's pasta.
So hello, everyone. Can you hear me? Perfect. So my name is Winnie, and I'm a graduate student at the Bazaki Lab. Today, I'm going to tell you recent projects on the role of shockwave ripples for selecting memory for consolidation during sleep. So there are a lot of words there. So let me start by unpacking 2 of the main things. First, what is shockwave ripples? Shockwave ripples are essentially synchronized patterns produced by extremely synchronized activity of neurons in the hippocampus. And they are an emergent population event, which means large number of neurons firing together in a temporally coordinated manner that cause this powerful population event called ripples. And ripples happens both during quiet awake and sleep. Another key word is selection. I'm going to tell you what this means and why this is interesting very soon. But let me start by asking this question to the members of the audience who did not sleep last night. Oh, wow, so some of you didn't sleep. All right, but sleep is really important. It's not only unavoidable, but it's also, our brains are also doing essential work while we slumber. Among many of the crucial tasks during sleep, there's this one on the agenda, which is to consolidate our memory during what we experience during awake. And we all know that we don't remember everything we experience during the day, because to remember, we forget. Only some of the memories are selected and consolidated during sleep, and those that get consolidated are hopefully get incorporated into the knowledge structure of the brain. So the essential question here is, how does the brain select what to consolidate during sleep. In order to have a more holistic picture of memory selection and consolidation during sleep, we need to go back and revisit this more basic question, which is, why does consolidation of memory take place during sleep? So the hypothesis that sleep serves memory consolidation is conceptually rooted in the two-state memory consolidation model. It assumes that memory are initially encoded in a fast learning store, which is the hippocampus. And what makes hippocampus a perfect candidate for this first state is that it forms fast associations, even in one attempt. But these representations are unstable and vulnerable for interference. Therefore, we need a second state. During second state, some of the new memories are gradually integrated into the slowly learning store, which is the neocortex. And those that are not consolidated are forgotten to free up space for encoding new memories. The key word here is, again, some. So with this foundation, we can go back to this fundamental question. How does the brain select what to consolidate during sleep? This is a complicated and big question, and I hope the project I will present today can give an important piece of the puzzle. So I will give you our answer right now. In short, we propose that shop with ripples during awake act as a memory tag, such that those memories that are tagged are consolidated during sleep. And those memories that are consolidated during sleep can be organized into the long-term storage permanently in the neocortex. In contrast, those that are not consolidated during sleep are forgotten. To iterate, awake ripples select what to consolidate, and sleep ripples consolidate them later during sleep. All right. Before I tell you our results, I want to first walk you through what is known so far to give you some important background information to understand our results. So I will introduce you the role of ripples, the known role of the ripples in memory consolidation for those in the audience that are unfamiliar. So as illustrated in the diagram, when animal navigates in the maze, the memory of this experience is encoded through the sequential firing of cells in the hippocampus. And later during sleep, there are those events called ripples that happens now and then. And what is really interesting is during some of those ripples, the exact same sequence that happens during memory encoding are reactivated again and again during ripples. And when this happens, we call those ripple events replays. So the relationship between ripple and replay is that only some of the ripples are replays. The replays are the ones that reactivate the sequence during the day. So we know that ripples are important for memory consolidation because without them, through loss of function manipulations, memory is impaired. So this is what we know. What does our new hypothesis add to this known picture? So our new insight here is that since not all memories are remembered, memories need somehow to be selected during or shortly after learning, and this memory selection mechanism is previously unknown. And we propose that awake ripples serve this function to select what to be consolidated later. So how do we study this question of memory selection in the lab? Let me start by introducing you to our task. So this is our task where this is called a figure 8 maze, where the animal runs left and right, in order to gain water reward here. And if then, so while animal doing this, we record the neuroactivity of CA1 region of hippocampus, while animal run many trials. Here we plot all the trial by trial trajectory and stack them together. After maze learning, we also record activity during sleep. Note that each trial is essentially an individual event. So we can begin to ask this question: can we differentiate those different trial events? So this is the question that we want to address in part one of the talk. And I want to remind you how does this part relate to our question at the beginning? Well, if we can differentiate those different trials, then we can have a handle to study how those different events are selected during sleep. So if we look at the trial by trial firing rate of some individual neurons, we get a hint that different trials are indeed encoded differently by some cells. For example, I'm showing one sample neuron. Its firing across the location of the track is color-coded by the trial number. So at first glance, you might think, oh, this cell has two place fields. But looking closer, you realize that this cell actually has different firing pattern across trials. At first, it fired at the middle of the track during early trials, and later switched its firing rate to the firing field to the end of the track during late trials. So this cell differentiates its pattern across different trial events. And this is another cell that are very silent during the early trials and only start to fire during late trials. And this is not due to electrode drift, because if we plot activity of exactly the same cell, but on the other side of the arm, we see this different pattern where the cells are firing both during early and late trials, but now at the left side, it fires more during early trials. So the picture we're having here is that at first glance, it seems like every trial you have a very similar firing pattern, but it's not quite. Some of the cells firing more during early trials, but decrease its firing rate during late trials. Some of, just like this neuron I showed you before, some of the cells have firing fields at the middle of the track during early trials, but switch its firing field to the late to the end of the track, just like this neuron. And there are also cells that firing only during the late trial. So you get the idea now. We know that individual neurons show this varying firing across trials. So the question is, if we have a lot of neurons and all of those different combinations of firing, and the question is if the unique combination among large population of cells is enough to reliably differentiate those trials. And we know that the individual neuron is fundamentally noisy. So this motivates us to harness the power of population analysis. So to do population analysis, we can use this method called dimensionality reduction. So briefly, we take the neuroactivity data while animals learn on the figure-8 maze, and our lab mate Roman can record up to about 500 neurons simultaneously. So we bin the data and smooth the data and do some pre-processing to have a spiking matrix like this. So note that because we have 500 neurons, this means this activity pattern leaving a 500 dimensional space, which is pretty high. So in order to find a structure within this very high dimensional space, we do dimensionality reduction with UMAP. So what we would see UMAP embeds the population activity to a low-dimensional space and output a structure like this. And we call this the neural manifold. I just want to remind you that each point on the neural manifold corresponds to a population vector in the original high-dimensional space. So each slice is a 100 millisecond thing, and this high-dimensional vector was embedded into the low-dimensional space and represented as a dot here. And what we see here is that this structure seems to capture the topology of the maze, which is pretty cool. Now that we have the structure that emerged purely unsupervisedly, we can take a closer look at the neural manifold. We can correlate the features of the manifold with various variables of the external world. For example, if we color the manifold according to the position of the animal, which is showing here, and to orange hue, the blue is the beginning of the track, and the green is where the animal gets rewarded, and the red is the end of the track. So what we see is a nice correspondence between the neural manifold and the topology of the maze. But this is very cool, but expected, given that we know there are place cells in the hippocampus. So what is really unexpected is if we color the neural manifold with trial number, which is shown here, I'm using the same color scheme, but the top is colored by position and the bottom is colored by trial number. We see this unexpected pattern emerge. So it seems like the neural representation not only tracks different positions, but tracks the progression of trial events. You can see this very structured activity pattern from early to late trials, where the representation of events that happens close to each other in time get embedded close to each other in the neural manifold. So what this means is that now we get a readout for the at the population level to differentiate different trial events. And now we want to move beyond visualization and to actually quantify this phenomenon. And one way to quantify this is to do decoding. So using semi-supervised dimensionality reduction with UMAP, we get a neural activity in low dimensional space like this. Semi-supervised here means that we get a unique label for every 5 trials in the data, such that the trials that get the same label are embedded very close together, just like trial 5125 here. And those that not share the same label are pushed far away from each other. And each ring here represents a unique trial block. And again, we see this nice progression from early to late trials organized in a very systematic way. And so UMAP gives us this very nice low dimensional space. On top of this, we can do decoding with K-nearest neighbor classifier, and we further validate our decoding results with tenfold cross-validation. And what we get is a confusion matrix like this. So as you can see in this confusion matrix, the probability mass really densely populate this area around the diagonal, which means predicted trial is most of the time consistent with actual trial. So we can decode this very reliably. We can further confirm this decoding result with 2 other methods, and the summary data is here. And interestingly, this phenomenon can be also observed in different tasks. In simple tasks like a linear maze, or more complicated maze like a radial arm maze. So the data on the left is recorded by Andreas Grossmark from a rat, and the data on the right is recorded by Thomas from a mouse. So this is nice because this means what we observe is a more universal phenomenon, not just restricted to the figure-8 maze. So now we can move on to the next part. So I've shown you that using dimensionality reduction, we can actually visualize population dynamics that reliably differentiate different trial events, and this can be decoded with 3 different methods. And also, this phenomenon is observed across diverse range of tasks and rodent species. And how does this relate to our selection question? Again, if we can differentiate those trial events reliably, later we can study how they get selected during sleep. So, but before we go to that, I want to dissect what we observe at the population level a bit further. So I showed you that we can decode different trials. Different trials can be decoded just means that they're sufficiently different. But the decodability doesn't tell us if this population activity is from random fluctuation or it's a structured variation. This is quite important question to answer because later we will study memory selection during sleep and do decoding. Before we move on to those complicated analysis, we want to make sure we're standing on the solid foundation. We want to make sure this activity pattern is structured. So how do we do that? In order to differentiate those 2 possibilities, random versus structured, I would first introduce you to this method called hold entire trial out cross-validation. So we start with a low-dimensional manifold by UMAP like this. And when you do cross-validation, you take some of the data out as testing data, and you get the rest of the data as training data. So here, instead of doing tenfold cross-validation, which is what we did before, now we specifically want to hold the entire trial out. So for example, we can hold trial block 4 out as testing data. As you can see, trial 4 is gone. And then we can use the rest of the data as training data to train our k-nearest neighbor decoder. And what we get, so trial number 4 will now be decoded to its closest neighbor. It cannot be decoded as number 4 because this is not an option. And the next adjacent trial is trial number 3. And this result can be summarized again in the confusion matrix. And then we see trial 4 has been decoded as number 3. This is different from tenfold cross-validation because the probability mass is not at the diagonal because this is not an option. But it nicely populates this area surrounding the diagonal, which really highly suggests this population activity is structured. And if we shuffle the data to perturb this, we see that the decoding error is very high. So I have shown you during part 1 and part 2 of the talk that there is a trial-by-trial variation in the hippocampal neural population pattern, and this variation is highly structured. We're now going to exploit this systematic variation in neurocode and use it to study the memory selection problem. I know I have shown you a lot of things, so take a breath because the most exciting part of the story is going to unfold now. But before I move on, I want to point out an interesting insight, which is why previous studies could not look into memory selection as what we do here. This is because in the past, when we decode the content of memory reactivation during ripple, most of the time, people build a template that is average across all the trials. And then later during sleep, you match the activity pattern during sleep to this average template. In contrast, what we can do now, courtesy of the technology, is that we can reliably differentiate each individual experience. And this technology is because at the hardware level, we can record large population of neurons simultaneously with this new probe. And we also have this powerful analysis tool to enable us to analyze this population data. So with this, this allows us to study the selection question. So moving on to the results, we will first focus on the awake ripples and then study the relationship between what is replayed during awake ripple and sleep ripples later. Okay, so when awake ripples happen, the first question we can ask is where they are. So if we quantify this, we realize that most of the awake ripples happen when and immediately after animal get rewarded. This is an example session on the left and summary of all sessions on the right. Now I'm going to show you an example awake replay event. This replay event took place shortly after the animal was rewarded, which is standard. And we can see that there's population burst during ripple and this very nice sequential reactivation of cells in the raster plot. Now, we can decode which part of the maze is being replayed by projecting the ripple event onto the maze manifold. So each time thing of a ripple event is represented by a purple dot here. And the number just indicates the sequence of the time being. Now that we know where those ripple events are at the position manifold, we can decode which part of the maze is being replayed. So this black triangle represents the physical location of the animal when this particular event takes place. The animal is here at the reward side. Just after it gets rewarded, it replays this part of the maze just ahead of the animal. So we can not only decode the content in terms of position, we can also decode which trial is being replayed. So again, triangle is the position, is the location of the animal. So animal is at trial number 7, trial block number 7, and the replay content is also trial block number 7, which means that the animal is replaying the current trial. So this is just an example event, but we can ask generally across all the awake ripples, do the awake ripples represent the past, future, or current trials? And this is the summary data. And we can see that most of the awake ripples replace the current trial. And another question related to trial we can ask is, are replay events evenly distributed across trials? And the answer is no. Some of the trials are replayed very frequently, and some of the trials are not replayed at all. So this information will become important later, so hold this in mind. Now we characterize the awake ripples. We can now move on to sleep ripples and see how events are selected to be consolidated during sleep replays. So what happens during sleep? So this event here is an example sleep event. As you can see, it is first of all, there's population burst during ripple, and this particular event during sleep is replaying about trial block number 5. And if we look at a similar event when animal is awake and on the maze, they look very similar. This is an awake replay when animal is on the maze, and it's replaying about trial number 5 as well. So which events are being selected during sleep replay? And one way to look at that is to plot the distribution in terms of trial that gets replayed during sleep. And we see this pattern. Some of the trials are replayed a lot during sleep, and some of the trials are less, and some trials are not at all. And what's really interesting is that this pattern mirrors the pattern during awake replay. So when we do the recording, we have the maze, we have the post-maze sleep. We also recorded sleep prior to the maze. This is act as a control, and as we can see, the pre-maze sleep replay does not have this correlated pattern that we see between awake and sleep. So this means this pattern, this trial reactivation pattern we see in post-sleep are experience dependent. And this is not only true in that one session, but across many different sessions. And if we do the summary data, we find a very high correlation between what is replayed during awake replay and what is replayed during sleep. So this means the trials that are replayed most during awake are also get replayed most during sleep. And to quantify this in a different way and comparing it with different variables that have been previously suggested could predict what's happening during sleep, we did this mixture effect linear regression analysis. Basically, this analysis is asking this question: which of those variables best predict what is the replaying pattern during post-sleep? And we can see that awake replays stand out. It is the best predictor for the trial distribution pattern during sleep. And we can confirm this with different methods. Okay, so what I'm showing you now is that to summarize and make sense for what I'm showing you is that between memory encoding and memory consolidation, there's this memory selection state. And our data suggests that what is being replayed during awake ripple, so the trial events that are replayed during the awake ripples get to be continuously replayed many times during sleep. So this is the take-home message summarized in this image. The awake Shopwolf ripples act as memory tags, such that memories that are tagged by them during or shortly after learning are selected. And then later during sleep, those selected memories are replayed time after time, many times during sleep. And those that are consolidated and replayed during sleep get incorporated into the existing knowledge base of the memories. And the non-selected memories are potentially forgotten. So this is the picture, and I hope there's awake ripple happening in the audience now, so that this picture is packed and you can remember this. So with this, I want to thank the authors of the project, without whom this project would be impossible. I want to also thank all of the current and previous Bazaki Lab members who gave me critical and constructive feedback all the way along. And I want to thank you, my thesis committee members and admin team. And finally, I want to thank you all taking your time during the middle of the day to attend my talk. Thank you. questions. Yes.
Hi, very interesting.